The protein organization of a red blood cell. Wisath Sae-Lee 1, Caitlyn L. McCafferty 1, Eric J. Verbeke 1, Pierre C. Havugimana 2, Ophelia Papoulas 1, Claire D. McWhite 1, John R. Hauser 1, Kim Vanuytsel 3, George J. Murphy 3, Kevin Drew 4, Andrew Emili 2, David W. Taylor 1, Edward M. Marcotte 1, 5. Highlights. The canonical RBC proteome consists of 1,200 proteins. These proteins form 100 complexes governing RBC structure, function, and dynamics. The band 3-ONC1 complex is a hub for RBC metabolic enzymes and cytoskeletal dynamics. Summary. Red blood cells, RBCs, erythrocytes, are the simplest primary human cells, lacking nuclei and major organelles and instead employing about 1,000 proteins to dynamically control cellular function and morphology in response to physiological cues. In this study, we define a canonical RBC proteome and interactome using quantitative mass spectrometry and machine learning. Our data reveal an RBC interactome dominated by protein homeostasis, redox biology, cytoskeletal dynamics, and carbon metabolism. We validate protein complexes through electron microscopy and chemical cross-linking, and, with these data, build 3D structural models of the ankyrin/band 3/band 4.2 complex that bridges the spectrin cytoskeleton to the RBC membrane. The model suggests spring-like compression of ankyrin may contribute to the characteristic RBC cell shape and flexibility. Taken together, our study provides an in-depth view of the global protein organization of human RBCs and serves as a comprehensive resource for future research. Graphical abstract. Download. Download high-res image, 326 kilobytes. Download. Download full-size image. Previous article in issue, next article in issue. Keywords. Red blood cells, erythrocytes, proteomics, protein complexes, band 3 complex. Research topics. CP, cell biology. Introduction. Red blood cells, RBCs, fill a critical role by transporting oxygen and metabolic waste between the lungs and other cells and tissues. This role has been optimized by the unique features of RBCs, which lack nuclei, mitochondria, Golgi, the endoplasmic reticulum, ER, and most other major organelles so as to maximize oxygen carrying capacity. Nonetheless, RBCs respond actively to changing tissue environments and dynamically alter their cell shapes on short timescales in order to thread narrow capillary networks and splenic tissues, pointing to the critical importance of protein interactions, allostery, and post-translational modifications as the likely primary mechanisms to regulate RBC activities. As a result, mutations that disrupt these aforementioned processes lead to various blood disorders, such as spherocytosis and hemolytic anemia. RBCs have been studied extensively for decades and have provided us with essential basic information on cell physiology and molecular biology. However, a consensus on their complete proteome has not been reached, nor is it known how these proteins organize into the large multi-protein assemblies that support all RBC function in the absence of transcriptional and translational activity. While hemoglobin comprises the vast majority of RBC protein content, 98%, Goodman et al. 2007, Cabanova et al. 2009, Pizzini et al. 2006, more than 1,000 other distinct proteins are predicted to constitute the remaining 2% of protein, many of which are still largely uncharacterized. Since proteins rarely act alone, building a more complete picture of multi-protein assemblies is key to better understanding RBC functions and diseases, including cell shape control and the shapeopathies that result from its disruption. Although techniques such as affinity purification mass spectrometry, APMS, and proximity labeling are available to study protein-protein interactions, PPIs, in other cell types, these approaches are not feasible in RBCs because of the lack of nuclei and transcription, and antibody-based approaches, such as immunoprecipitation MS, IPMS, are prohibitively expensive for large-scale screening. Therefore, we turn to another powerful technique, to study protein complexes, co-fractionation mass spectrometry, CFMS. CFMS is a high-throughput technique that combines biochemical fractionations, protein MS, and machine learning to characterize PPIs, in which the co-elution, co-fractionation, of stably associated proteins through the course of distinct and orthogonal biochemical separations serves as evidence for the protein's physical association. Skinnider and Foster, 2021, Wan et al., 2015. As CFMS requires neither antibodies nor recombinant epitope tagging of individual proteins, it is uniquely well suited to study RBCs. The power of this technique comes from the analysis of native proteins' elution profiles from multiple orthogonal biochemical separations using machine learning and extensive internal control proteins, known complexes, to distinguish between true PPIs and randomly co-eluting proteins, quantifying the support for the physiologically relevant PPIs in order to maintain strong control over false discovery rates of protein interactions. In this work, we performed over 30 biochemical fractionations on purified RBCs and applied quantitative MS on these fractions, greater than 1,900 MS experiments in all, in order to identify a canonical set of approximately 1,200 RBC proteins and to derive an interactome map of RBC protein complexes. Using a rigorous statistical framework, we recovered high-confidence PPIs from known complexes as well as novel complexes. Most of the complexes in RBCs are involved in energy metabolism, structural integrity, redox biology, and proteostasis. Furthermore, we performed chemical cross-linking on these native complexes and used integrative structural modeling to shed light on the molecular organization of integral membrane proteins, channels, cytoskeletal proteins, and metabolic enzymes at the RBC membrane. Our findings provide a comprehensive blueprint of the cell surface and subcellular architecture of a key blood cell type and suggest biophysical mechanisms underlying its adaptability and pathological reorganization in diverse blood disorders. Results and discussion. A large dataset of protein abundances and co-purification of RBC proteins. In order to characterize the RBC proteins, Figure 1 and their assemblies Figure, we generated a large proteomics dataset of fractionated soluble and membrane-associated proteins from enriched human RBCs using various methods of biochemical fractionation. Non-denatured protein extracts from hemolyzate, soluble proteins, and non-ionic detergent-dissolved ghosts, membrane proteins, were separated by discrete biophysical properties such as size, charge, and hydrophobicity. See metadata.xlsx in the Zenido data repository for details of fractionations, donors details, and detergents used. Each chromatographic fraction was analyzed by high-resolution, high-sensitivity liquid chromatography (MS) (LCMS). In all, we collected 6,255,027 interpretable peptide mass spectra from 1,944 individual chromatographic fractions. Each fraction captures distinct subsets of native RBC proteins and protein assemblies, collectively providing a compendium of informative co-elution profiles. Download. Download high-res image, 1 megabyte. Download. Download full-size image. Figure 1. Defining the canonical red blood cell, RBC, proteome, with high accuracy from a synthesis of protein mass spectrometry and mRNA expression data. A. Measured protein and RNA abundances from diverse blood cell types and plasma were used as features for a machine learning classifier to assign confidence scores for proteins, whether they belong to the RBCs or rather contaminants contributed by other cells or plasma. The classifier showed high precision and recall. Area under the recall precision curve equals 0.97 as assessed on a 172 protein set withheld from the training. Cell images created with biorender.com. B. Applying the classifier and thresholding at a 1% false discovery rate, FDR, we observed the canonical RBC proteome to comprise 1,202 proteins. The resulting high-confidence RBC proteins are highly concordant with proteins previously identified from purified erythrocytes, Gautier et al., 2018. In contrast, the 1% FDR proteome notably excludes proteins known to be strongly enriched in reticulocytes, Gautier et al., 2018. D. To further assess the potential for proteins to be contributed from other blood cells or serum, we differentiated iPSCs into polychromatic and orthochromatic erythroblasts in serum-free medium lacking white blood cells, platelets, and serum proteins, and then analyzed the erythroblast proteome using mass spectrometry. E. A large majority of high-confidence RBC proteins at both the 1% and 5% FDR level, 1,202 proteins in total, were also detectable in erythroblasts, consistent with our expectation that mature RBC proteins should generally be detectable in a relevant precursor cell population. Download. Download high-res image, 3 megabytes. Download. Download full-size image. Figure 2. Overview of the integrative co-fractionation of mass spectrometry, CFMS, workflow used to determine stable RBC protein complexes. A. Hemolysate and white ghosts are chromatographically separated, and the proteins in each fraction are identified by Missolution profiles for each protein are graphically represented as ridgelines across multiple separation experiments. Cell and protein complexes images created with biorender.com. B. Different measures of correlation between each pair of proteins are used to construct a feature matrix for machine learning, which computes a score, CFMS score, indicating how likely the interaction between two proteins would be in RBCs. The classifier showed good precision and recall. Area under the recall precision curve equals 0.45 as assessed on 299 PPIs withheld from the training. Further validations were achieved through cross-linking MS and direct visualization by electron microscopy. C. Heat map of the full dataset of abundance measurements for each of the 1,202 RBC proteins across all fractionations of hemolysate and white ghosts. Blue indicates non-zero signal. D. Enlarged portions of C, showing examples of strong coelution observed for subunits, gene names on left, of 5 well-known protein complexes in RBCs, complex names on right. Color intensity, blue is positive signal, depicts abundances for each protein from a representative hydrophobic interaction chromatography experiment labeled on top out of the 30 total separations. Determination of a high-confidence RBC proteome. From this large dataset, we first aimed to comprehensively define a canonical set of RBC proteins. Although multiple previous surveys have identified versions of the RBC proteome, Brick and Wisniewski, 2017, Goodman et al., 2007, Lang et al., 2014, these studies only agree on 859 constituent proteins, Figure 1a. Discrepancies might arise from technical variation in the analyses and samples, as well as from the presence of contaminating proteins contributed by accompanying reticulocytes, platelets, white blood cells, WBCs, and serum proteins. We attempted to reconcile these published datasets and our own data using a bioinformatic approach. We trained a machine learning classifier on available MS and RNA sequencing, RNA-seq data from different blood cell types, Figure 1a, in order to recognize the best supported consensus set of RBC proteins. The rationale for this approach is that some proteins detected in previous studies or our fractionation experiments inevitably derive from non-RBC blood cells, despite best efforts to enrich RBCs. This issue is exacerbated by the fact that all non-hemoglobin proteins only represent 2% of the RBC proteins, and thus even 2% of contaminant cells could skew protein identifications for RBC proteome. The consideration of data spanning multiple cell types should allow RBC proteins to be better distinguished from proteins from other blood cells and serum. There are two major issues to consider when trying to identify the comprehensive list of RBC proteins: coverage and contamination. In terms of coverage, we detected more than 90% of the highly confident set of 859 previously described proteins among the 2,000 most abundant proteins from our experiments, Figure S1a. This analysis shows that our co-fractionation experiments were well powered to detect most RBC proteins. Some exceptions are notable, particularly alpha and beta actins. and active, which are systematically depleted due to our use of mild and non-ionic detergents to extract proteins while preserving stable interactions, and the Duffy antigen ACKR1. In order to distinguish bona fide RBC proteins from other cell types, we employed a supervised machine learning approach to quantify the likelihood of proteins deriving from RBCs. We analyzed protein and RNA abundances from MS and RNA-seq data from RBCs, RBC precursor cells, other blood cell types including WBCs, platelets, and reticulocytes, and serum, Figure 1a, with a derived likelihood score based solely on these measurements. As confirmed positive training examples, we selected a set of 687 known RBC proteins out of the 859 proteins agreed upon by all three reference studies, and we withheld the remaining 172 positive examples as an independent test set. As confirmed negative training examples, we considered all human proteins observed in our proteomics experiments that were not observed as RBC proteins by the three prior studies. We then trained a random forest classifier, with five-fold cross-validation, to assign a confidence score between 0 and 1 to each protein. See RBC interactome pairwise interactions.xlsx in the Zenido data repository for scored proteins, with 1 indicating a high likelihood of the protein existing in mature RBCs and 0 indicating a likely contaminant. We assessed the quality of these confidence scores by comparison to the withheld test set of 172 known RBC protein markers and a set of withheld negative test examples. The classifier performed extremely well, as indicated by a high area under the precision recall curve, AUPR, of 0.97, Figure 1a. Ranking proteins based on their likelihood scores enabled us to measure classifier false discovery rates, FDRs, Figure 1b. At a likelihood score greater than 0.55, we observed 1,202 proteins at 1% FDR, Figure S1b, all of which were directly supported by proteomics evidence. See RBC proteome and abundances.xlsx in the Zenido repository. We consider this stringent threshold to define a comprehensive and high-confidence set of RBC proteins. Of the 859 Goldstandard proteins, 785 are recovered in our canonical RBC proteome at 1% FDR, and 26 more recovered at the 5% FDR rate, leaving 48 with a substantially lower degree of support from all sources of evidence included in our classifier for being true members of the RBC proteome. Ranking the proteins detected in each of the three prior RBC proteome studies by our RBC likelihood score shows that our classifier efficiently distinguished RBC proteins from non-RBC proteins, figures S1c, S1e. Among the reference studies, the preparation by Lang et al. 2014 comprised a notably high proportion of mature RBCs. Besides well-established RBC proteins, such as hemoglobin, carbonic anhydrase, RBC-specific membrane proteins, for example band3, ankyrin1, and band 4.2, and blood-type antigens, for example Rh antigens and KEL, the high-confidence RBC proteome faithfully reflects the unique biology of mature RBCs in that they lack major organelles or the apparatus for DNA replication, transcription, and protein synthesis. To further support the proteome, because most proteins in mature RBCs would be already present in RBC precursor cells, we asked whether they were additionally present in induced pluripotent stem cell, derived erythroblasts. While nucleated, these RBC precursors were grown in serum-free medium, Vanuytsel et al. 2018, and were devoid of all contaminant proteins from other blood cell types and from serum, Figure 1d. In all, 83% of the high-confidence RBC proteins in our compendium, 1% FDR, were also present in the erythroblasts. Similarly, we compared the high-confidence set of RBC proteins with those obtained through multi-step RBC purification, including centrifugation, gradient and cellulose purification, and fluorescence-activated cell sorting, FACS, by Gauthier and colleagues, Gauthier et al., 2018. The majority of RBC-specific proteins obtained in this way exhibited high RBC likelihood scores, while the majority of proteins obtained from similarly enriched reticulocytes had markedly lower RBC likelihood scores, Figure 1c. Indeed, the high-confidence RBC proteome excluded CD71, transferrin receptor protein 1, TFRC, a well-known reticulocyte marker protein, Gautier et al., 2016-2018, Goodman et al., 2007. Taken together, these results suggest that our bioinformatic approach effectively captured the proteome of mature RBCs and excluded most major contaminating non-RBC proteins. Interestingly, some proteins with high RBC likelihood scores consisted of annotated subunits of ribosomes, nuclear transport proteins, and parts of the ER in Golgi machineries, despite RBCs lacking these larger systems. Many of these proteins were also detected as RBC-specific populations by Gautier et al., 2018, and were observed in the EBSC-derived erythroblasts. Thus, they are unlikely to be contaminants. Although we cannot rule out the possibility of moonlighting functions in RBCs, another explanation may simply be that such proteins are not eliminated or degraded effectively during the process of enucleation in reticulocytes, in which the nucleus was expelled, major organelles were eliminated, and other superfluous proteins were degraded. Thus, the proteome could at least in part reflect an imperfect cell maturation process. Systematic identification and scoring of stable RBC protein interactions. Given this high-confidence set of RBC proteins, we next sought to determine their association into higher-order multiprotein assemblies. In CFMS, subunits of many well-known complexes coalute with distinct patterns across different types of biochemical separations due to differences in complex sizes, charges, and chemical properties, Figure 2a. From our experience working with different tissues and organisms, detergent-dissolved membrane protein complexes, such as the band-3 complex in Figure 2c, tend to have broader chromatographic peaks compared with discrete soluble complexes, such as the proteasome and CCT complexes. Membrane proteins often exist in a variety of forms with different amounts of detergent, lipids, and interaction partners, resulting in their detection across multiple biochemical fractions. To analyze such differences and identify co-eluting proteins in a systematic and high-throughput manner, we again used a supervised machine learning approach in order to assign a confidence score to each potential PPI based on the MS evidence, Figure 2b. Importantly, PPIs were derived solely from the separation behavior of the observed proteins over multiple orthogonal biochemical fractionation experiments directly from RBCs, with no contribution from external data, see STAR methods and the Zenodo data repository. We focused on abundant RBC proteins with the most robust MS evidence, more than 60 independent peptide spectral matches, PSMs, across 1,944 biochemical fractions. We trained an extra tree classifier, using five-fold cross-validation, to distinguish between pairs of proteins known to stably interact and random pairs of proteins, details in STAR methods. The classifier assigned a probabilistic CFMS score between 0 and 1 to each potential PPI, with 1 indicating a high likelihood of physical association based on observing strongly coordinated protein elution profiles, and 0 indicating no evidence for interaction in RBCs. We judged the quality of the measured interactions by comparing with a withheld test set of 299 known protein interactions, Figure 2b, which allowed us to measure classifier error rates. The CFMS scores accurately recapitulated the test PPIs. For interactions with CFMS scores over 0.17, we observed 85% precision and 35% recall, Figure 2b, with an OPER of 0.45, well above the 0.03 expected by random chance. In all, we observed 3,229 PPIs among the high-confidence RBC proteins that scored 0.17 or better, providing an estimate of the RBC interactome. Direct visualization of biochemically fractionated complexes by electron microscopy. Because CFMS involves biochemical enrichment and separation of intact endogenous protein complexes, we could use electron microscopy (EM) to directly visualize the larger complexes in a relatively unbiased fashion. This combination of TFMS with EM on cell extracts can provide rich data for near-native structure determination. Castritis et al. 2017, Kirilis et al. 2021, McCafferty et al. 2020, Verbeke et al. 2018, and has been applied to diverse samples. Aramura et al. 2021, Kim et al. 2020, Kirikowicz and Woodward 2020, Yi et al. 2019, including membrane proteins, Su et al. 2021. First, to survey the size, shape, and complexity of assemblies across a representative biochemical separation, we performed negative stain EM on pooled adjacent fractions from a size exclusion chromatography separation, Figure 3a. By comparing the micrographs with proteins identified in the corresponding MS experiments and prior knowledge of 3D structures, we identified 4 distinct protein complexes across the fractions, Figure 3b, 3 of which were large homo-oligomeric assemblies. Our ability to identify MS observations with EM particles was largely due to the lower complexity of the RBC proteome, which is 1/4 the complexity of E. coli cells, but is dominated by a smaller number of high-abundance components that are the major proteins in the MS and EM. The tripeptidyl peptidase 2, TPP2, was the largest observed homo-oligomer and is known to form 5 to 6 MDA assemblies, McPherson et al., 1987, Schoenig et al., 2012. The other homo-oligomers identified were the porphyrin biosynthetic enzyme delta-aminolevulinic acid dehydratase, ALAD, Mills-Davis et al., 2017, and 290K-de homo-octamer with D4 symmetry, and the peroxyredoxin PRDX2, Schroeder et al., 2000, and 218K-de homo-decamer with D5 symmetry. Although the EM reconstructions are at relatively low resolution, known protein structures could be readily docked into the EM reconstructions, confirming their identities and structural integrity throughout the separations. Download. Download high-res image, 3 megabytes. Download. Download full-size image. Figure 3. Validation of the TFMS workflow using electron microscopy confirms intact multi-protein complexes. A. Hemolyzate from size exclusion chromatography was partitioned into 5 groups and visualized with negative stain EM. Elution profiles from corresponding MS data were used to assist in identifying abundant protein assemblies. B. Reference-free 2D class averages of 4 protein complexes spanning 220 to 5000 kDa were identified in hemolyzate. C. Cryo-EM reconstruction of the 20S proteasome. Negative stain structures of TPP2 and PRDX2, along with docking of their corresponding atomic structures PDB, 3LXU, Chuang et al., 2010, and PDB, 1QMV, Schroeder et al., 2000, respectively. These data also clearly showed a high abundance of proteasomes, which provided an opportunity to revisit an outstanding question as to whether proteasomes in mature RBCs are active, i.e. in their regulated 26S forms, or mostly inactive, with separated 20S cores and 19S caps. We first pooled the proteasome-containing fractions 10 to 21, for higher resolution analysis by cryo-electron microscopy, cryo-EM. By single-particle analysis, we obtained a reconstruction of the 20S proteasome directly from RBCs with a nominal resolution of 3.35 A ring, confirming an intact, not visibly modified core proteasome, figures 3C and S2. To further investigate the proteasome's activity in RBCs, we used negative stain EM to survey hemolyzate after being passed through a 100K Differ filter and quantify different assembly states of the proteasome, figure S3. We found that 94% of the proteasomes observed were of the free 20S form, while the remaining 6% were singly capped 26S proteasomes, figure S3. These results agreed with our CFMS observations of generally separated 19S and 20S proteasomes and suggested that active 26S proteasomes were present only in relatively low abundances, although we cannot rule out the possibility of the initial cell lysis and hemoglobin depletion processes altering the proteasome assembly states. The interactome of mature red blood cells. The visualization of known complexes by EM confirmed that stable multiprotein assemblies were preserved well across our biochemical separations, so we next sought to systematically define protein complexes in mature RBCs by clustering the proteins based on the measured pairwise interactions. To provide a more comprehensive view of protein complexes, we elected to use multiple clustering cutoffs to reflect the hierarchies intrinsic to interacting proteins, the results of which are visualized in Figure 4. Download. Download high-res image, 670 kilobytes. Download. Download full-size image. Figure 4. A map of the primary RBC multiprotein complexes. Thin circles show the clustering hierarchy of protein-protein interactions into complexes for each of 3 clustering thresholds. See RBC_interactome.xlsx in the Zenodo data repository for complex memberships and annotations. Proteins, filled circles, are colored by broad biological categories, analyzed using the David annotation tool. Bold outlines denote proteins with oxidoreductase activity. In all, the interactome of mature RBCs represents a remarkably compact and minimal assembly of protein machinery. First, we find just over 100 protein complexes in mature RBCs. While this is already apparent at the proteome level, 1,200 total proteins in mature RBCs, or approximately 1/4 the complexity of the proteome of the bacterium E. coli and 1/16 of the consensus human proteome. The number of complexes is still striking in comparison to the greater than 400 complexes in E. coli, Caulfield et al., 2015, Hu et al., 2009, and greater than 7,000 human protein complexes known to date, Drew et al., 2021. To gain some insights into the interactome, we analyzed the proteins according to their broad functional categories, Huang et al., 2009a, 2009b, and found most categorized proteins mapped into 4 major categories: proteostasis, cytoskeleton and structural integrity, energy, and others. The clustering evident in the map supports the tendency to correctly associate proteins that bind each other and shows the relative balance of the different functions across the interactome. Proteins of proteostasis dominate the observed RBC interactome, notable among these being the 19S regulatory cap and 20S core of the proteasome. However, consistent with the evidence from EM micrographs, Figure S3, the clustering of PPI suggests that proteasomes in mature RBCs generally exist as distinct 19S and 20S forms. While the 26S proteasome is essential for the enucleation and maturation process, as RBCs remove many of their precursor proteins and maximize cellular space for hemoglobin, it is unclear whether proteasome activity is essential in RBCs post-maturation. Our detection of higher amounts of the 19S and 20S forms than the 26S form suggests that canonical ubiquitin-dependent degradation might be kept at a minimal level in mature RBCs, since this process requires the 26S form. However, it is possible that the 20S proteasome may still act to degrade unfolded and misfolded proteins without the 19S cap, especially when disordered regions are exposed, as seen in a few reports. Alvarez-Castelao and Castaño, 2005, Asher et al., 2005, Sorokin et al., 2005. In addition, the prevalence of 20S and 19S forms over 26S might also result from a more oxidizing cellular environment in RBCs than other cells, such as muscle cells, 11:1 ratio between NAD+ and NAD for muscle cells and 8:1 for RBCs, Demarest et al., 2019. NAD is known to bind to at least 5 19S subunits and stabilize the 26S form in turn, Tsvetkov et al., 2014. While we observe many proteins related to proteostasis, some of the proteins that fall into this category include chaperones and ubiquitin-related enzymes, which might be remnants of the robust translation and active ubiquitin-dependent degradation during maturation. Besides proteostasis, energy production and redox-related protein complexes were abundant. A survey of the interactome suggests a balance in regulating energy production and managing toxic byproducts through detoxification and redox enzymes, bolded circle, Figure 4. RBCs consume energy to maintain proper ion concentrations and appropriate ratios of surface area to cell volume, Lux 2016, Mohandas and Gallagher, 2008, which are essential to the ability of RBCs to change morphology without lysing. The major source of ATP production in RBCs involves the metabolism of glucose via the Embden-Meyerhof or glycolysis pathway, Brown, 1996. One of the toxic byproducts is methylglyoxal, a highly reactive dicarbonyl compound that reacts with proteins through the Maillard reaction to form glycated proteins, impairing protein function. In order to detoxify methylglyoxal, cells rely on the glyoxylase system and complex, which is recapitulated in the interactome. We find evidence for interactions relevant to methylglyoxal detoxification occurring between triose phosphate isomerase, TPI1, and the Parkinsonism-associated protein, DJ1/PARC7. TPI1 is known to produce a significant amount of methylglyoxal as a byproduct, Richard, 1991, and PARC7 is proposed to be a bona fide deglycase, Richarm and Deru, 2017. This TPI1-PARC7 interaction suggests a physical linkage between energy production and byproduct detoxification in RBCs. Finally, cytoskeletal complexes are essential to RBCs, as they maintain structural integrity and enable cell morphology changes as RBCs traverse blood vessels. The interactome recapitulates well-known cytoskeletal complexes involving, among other proteins, spectrin, adenosine-dermatin, and band-3, and suggests subcomplexes such as band-3, GLUT, and band-4.2, as described in more detail below. Cross-linking and integrative 3D modeling of the major RBC cytoskeletal complexes. As cytoskeletal complexes are essential to many RBC functions, we particularly sought to investigate protein complexes isolated from RBC white ghosts, which are composed almost exclusively of membrane and cytoskeletal components. Thus, we separated detergent-solubilized white ghost protein extracts by size exclusion chromatography and performed chemical cross-linking MS-XL-MS in order to precisely determine amino acid resolution PPI contacts between membrane and cytoskeletal proteins. We chose disoxynimetyl sulfoxide, DSSO, for cross-linking, as it is MS-cleavable and thus suitable for highly accurate determination of cross-linked peptides, Cao et al., 2011. In all, we identified 769 high-confidence crosslinks among 129 proteins, dominated by interactions among spectrins, ankyrin, and their interaction partners, including the Rh antigens, band 3, and band 4.1, figure 5a. Cross-linked proteins were more likely to have high MS scores, suggesting that the experiment captured true molecular interactions, figure S4. In addition to intermolecular crosslinks, figure 5a, we observed large numbers of intramolecular crosslinks, as highlighted in figure 5b for spectrin alpha and beta. Download. Download high-res image, 2 megabytes. Download. Download full-size image. Figure 5. Chemical crosslinks confirm mapped interactions and constrain 3D modeling of membrane and cytoskeletal complexes. A network plot represents cross-linked interactions among proteins, shown as gene names, in RBC cytoskeletal complexes. Line between each node indicates detected crosslinks. Shaded lines indicate dense crosslinking. B. Bar plot shows extensive crosslinks between spectrin alpha and beta, supporting a head-to-tail conformation. Numbers under each bar indicate the amino acid position on each protein. Yellow line indicates intramolecular crosslinks, and purple line indicates intermolecular crosslinks. C. Side view of integrative structure of band 3 ankyrin 1 complex and band 3 GIPA complex. Our model suggests that GLUT competes with the Rh proteins for binding with band 3 and band 4.2. The outline figure on the right shows intramolecular and intermolecular crosslinks that are overlaid onto the structure. D. Top view of integrative structure of the band 3 ankyrin 1 complex and band 3 GIPA complex. E. Integrative structure of band 4.1 spectrin complex. 6 of these complexes are proposed to link spectrin heterodimers with actin, Lux, 2016. Each crosslink generally provides evidence for the modified lysines residing within 30 ring of each other. Thus, such data enable initial molecular models to be constructed of the component proteins. For example, the pattern of crosslinks between spectrin alpha and beta strongly supports them associating in a head-to-tail conformation, figure 5b, consistent with literature reports that the N-terminal domain of spectrin alpha interacts with the C-terminus of spectrin beta. Spiker et al., 1992. Unjwickel and Gretzer, 1978. To further evaluate the quality of the crosslinks, we used known X-ray crystal structures of the proteins band 3, PDB, 1-hen and 4-YZF, glyceraldehyde 3-phosphate dehydrogenase GAPI, PDB, 1U8F, and phosphoglycerate kinase 1, PGK1, PDB, 4033, to calibrate the crosslinks accuracies. Of the 24 crosslinks between residue pairs within these X-ray crystal structures, all 24 occurred between amino acids with C-alpha atoms falling within 30A ring of each other, consistent with the length of the DSSO crosslinker, figure S4. Next, we integrated the crosslinks with available structural information in order to build models of several of the major RBC membrane and cytoskeletal complexes. We additionally incorporated knowledge of the positions of transmembrane regions, the UniProt Consortium 2019, protein structures from comparative modeling, and, where available, X-ray crystal structures. Using integrative modeling, figure S5, Russell et al., 2012, we built many 600,000 models in parallel and tested for their agreement, C-star methods, the combination of annotated transmembrane regions, chemical crosslinks, and known partial structures provided sufficient spatial restraints to converge on a solution. High-scoring models that satisfied the cross-linking and membrane restraints were clustered based on root mean square distance, RMSD, and the largest cluster, table S2, was selected as the preferred form of each complex, with the centroid model as the representative model for the cluster. This integrative modeling approach provided us with a detailed molecular view of both the band 3, figures 5C and 5D, and band 4.1 complexes, figure 5E. Yellow and purple lines to the right of the models in figures 5C, 5E highlight positions of intramolecular crosslinks and intermolecular crosslinks respectively. Surprisingly, 30% of the crosslinks for ankyrin 1 violated the 30A ring distance of the crosslinker, far in excess of the typically fewer than 20% of violations expected in such models. Leitner et al., 2016, Liu et al., 2018, Wang et al., 2017, and the 100% concordance we observed with the isolated band 3 and PGK1 structures, suggesting that the reported X-ray crystal structure of ankyrin 1, Wang et al., 2014, does not match the predominant form found in RBCs. Notably, the reported structure is in an extended, open form. Our crosslinks suggest that the violation could be due to ankyrin adopting a compressed, closed conformation, as the violations are highly directionally correlated in two specific regions of ankyrin, figure S6. When modeling a closed conformation of the ankyrin complex, 92% of the crosslink violations for ankyrin 1 can be satisfied, figure S6. We speculate that the open and closed conformations could be a result of spring-like behavior of ankyrin repeats. Indeed, spring-like behavior has been previously observed for ankyrin by atomic force microscopy. Lee et al., 2006. Given ankyrin's privileged position connecting the RBC plasma membrane via interactions with the band 3 membrane proteins to the spectrin cytoskeleton, it is plausible that spring-like behavior of ankyrin may play an important role in maintaining RBC's morphologies as they deform while traversing microvasculature and splenic tissues. Indeed, the band 3 ankyrin 1 complex has been proposed to locate in the middle of the spectrin tetramer chain to anchor spectrins to the plasma membrane, while the band 4.1 complex connects the end of the tetrameric spectrin chain to one of 6 binding sites on actin in the actin junctional complex. Lux 2016, Mohandas and Gallagher 2008, figure 6a. Our model of the band 3 ankyrin 1 complex shows the association of band 3 with band 4.2, R8 proteins, GLUT, ankyrin, spectrin, and glycophorin A. The association of band 3 with band 4.2 in GLUT based on cross-linking corresponds well with the cluster derived from CFMS, figure 4. In addition, our model of the band 3 ankyrin 1 complex is supported by previous co-immunoprecipitation data from RBCs of an individual with almost complete absence of band 3, Coimbra mutation, homozygous null for band 3, crystallography, in vitro binding assays, and M.S. Bruce et al., 2003, Ipsaro and Mondragon 2010, Ipsaro et al., 2009, Zhang et al., 2006, Kompornsen et al., 2011, Lemon et al., 1992. A previously observed reduction of Rh proteins and band 4.2 in the absence of band 3 suggests that they also form a complex in RBCs, Bruce et al., 2003. Interestingly, Bruce and colleagues found that GLUT was detected more in the absence of band 3. Our cross-linking evidence and 3D model indicate that GLUT competes with Rh proteins at the same binding sites and regions on band 3 and band 4.2, while band 3 and glycophorin A form a separate complex. These data thus support at least 3 subpopulations of band 3 complexes, those with Rh proteins, GLUT, or glycophorin A. Download. Download high-res image, 1 megabyte. Download. Download full-size image. Figure 6. Reconstructions of band 3 ankyrin 1 accessory protein complexes by integrative 3D modeling suggest ONC1 compression links the membrane to the cytoskeleton. A. An overview of the cytoskeletal network supporting the membrane of RBC. A pseudohexagonal network of spectrin heterotetramer underlies the membrane and is anchored to the membrane by the band 3 ankyrin 1 complex. The tetramer is attached to actin on the other end through the association of band 4.1, actin, spectrin, and other proteins. An actin polymer can interact with 6 spectrin tetramers through band 4.1, Lux 2016, adapted with permission from Goodman 2020. B. Glycolytic enzymes, such as GAP and PGK1, are anchored to the band 3 ankyrin 1 complex, which can accommodate these enzymes, while ONC1 adopts either open or closed conformations. See IMP_supplement_ in the Zenital repository for more details. ONC1 in an open form in the band 3 ankyrin 1 complex. ONC1, GAP, and PGK1 are colored. D. Roughly 1/3 of observed intramolecular ONC1 crosslinks supported adopting a closed form in situ relative to the extended conformation observed for purified ankyrin, Wang et al., 2014, suggesting that ONC1 is capable of adopting either open or closed conformations. See IMP_supplement_figures in the Zenital for more details. In addition to the membrane and cytoskeletal proteins, we found cross-linking evidence for glycolytic enzymes associating with the band 3 ankyrin 1 complex. Based on our modeling, both the potential open and closed forms of ankyrin in the band 3 complex, figures 6b/6d, can accommodate GAP and PGK1 binding without any violations of cross-linker distances. Thus, the positions of these enzymes with regards to the band 3 ankyrin 1 complex do not seem to have an impact on the structural integrity of the complex as a whole. Previous experiments confirmed the association of these two enzymes with the RBC membrane. It was found to be associated at the red cell membrane via immunofluorescence and binding assays, Rogalski et al., 1989, and PGK1 activity was detected in RBC white ghosts, Harris and Windsor, 1990, Ronquist and Ogren, 1966, Schreier, 1966. It is possible that localizing energy-producing enzymes to the RBC membrane where ion channels, transporters, and cytoskeletal network are located could be a way for RBCs to rapidly adjust the ratio of cell volume to surface area and change conformational states of cytoskeletal proteins such as ankyrin and spectrin in a timely manner. In turn, these rapid changes would facilitate dynamic morphological changes in response to the movement of RBCs through tissues and microvasculature. The structure of the band 3 ONC1 complex has recently been investigated using cryo-EM and reported in 2 preprints, Valls et al., 2022, Xia et al., 2022. Although the underlying structures have not yet been deposited, the structures are strikingly similar to our integrative model based on cross-linking and transmembrane domain restraints in that band 3 directly binds, i.e. shares a contact interface, with both band 4.2 and ankyrin 1. Consistent with Valls et al., we find that band 4.2 specifically interacts with the N-terminus of band 3, with the N-terminus of band 4.2 oriented to the membrane. Consistent with Xia et al., we find ankyrin repeats 6 to 13 directly contact band 4.2. Most notably, multiple variations are evident for the composition of the complex. 1. Valls et al. Observed interactions of the band 3 ankyrin 1 complex to aquaporin 1, which we also detected by cross-linking. However, the number of crosslinks were insufficient for modeling. 2. The cryo-EM structures from both groups of the band 3 ankyrin 1 complexes lack spectrin, a known interactor, Epsaro and Mondragon, 2010, Epsaro et al., 2009, and a key component of our model supported by 7 unique intermolecular crosslinks between spectrin alpha and beta and band 3 ankyrin 1. We speculate that the absence of spectrin could account for these authors observing open ankyrin conformations rather than the closed conformation evident in our data, figure S6. 3. Gludi is absent from these cryo-EM structures. In our model, the presence of gludi is supported by both TMS and 2 independent high-confidence crosslinks. 4. A variety of alternate subcomplexes are observed by all 3 groups, likely resulting from the different methods of fractionation employed, size exclusion then crosslinking versus glycerol gradient, and salt conditions, similar salt concentrations between our work in Valls et al., 2022, versus low and high NaCl exclusively in Xia et al., 2022. At a minimum, it is clear that the band 3 ankyrin 1 complex possesses multiple, heterogeneous assembly states. At the junctional complex, actin, non-muscle myosin, and other proteins work together to assert force on the spectrin network and thereby facilitate RBC deformability, Smith et al., 2018. At least under mild detergent conditions, the cross-linking and modeling of band 4.1 and spectrin suggest a stable interaction in the absence of actin. However, we note that the interactions may vary when actin is included, as seen in the example of band 3 ankyrin 1 complexes isolated using different salt concentrations or separation methods, Valls et al., 2022, Xia et al., 2022. Other techniques, such as super-resolution microscopy, might be able to shed more light on these complexes in their native conditions. Taken together, the band 3 ankyrin 1 complex maintains structural integrity by linking the spectrin network to the membrane while also serving as the hub for metabolic activities. These metabolic activities include energy metabolism, glycolytic enzymes, and ions and gas exchange, ammonium through RH proteins and CO2, HCO3-, Cl- through band 3, and carbonic anhydrase, Sterling et al., 2001. Such elaborate organization of channels, integral proteins, and enzymes makes RBCs a unique and minimalist system that can undergo rapid morphological changes while preventing cell lysis from occurring. Conclusions. We have used protein MS/EM, chemical cross-linking, and 3D modeling to systematically determine the protein organization of the simplest primary cell in the human body, the erythrocyte. After defining a consensus set of canonical RBC proteins, we constructed a reference map of RBC protein interactions and multiprotein complexes, providing a detailed look at the molecular organization of RBC proteins. A 3D model of the band 3 ONC1 complex provides amino acid level detail of a critical protein complex that underlies one of RBC's most fascinating features, the ability of RBCs to undergo rapid dynamic morphological changes, a key aspect of controlling cellular form and activity given RBC's absence of gene expression regulation. The detailed molecular organization of proteins in the band 3 ankyrin 1 complex clarifies the structure of a major membrane cytoskeletal attachment site, supports the possibility of spring-like behavior at the membrane and cytoskeleton connection, and suggests aspects of RBC metabolism are spatially organized. In all, these data represent the near-complete map of PPIs in any primary human cell type, and thus provide an essential reference for these minimalistic cells, laying the groundwork for future full-cell molecular models. Limitations of the study. Despite the extensive characterization of the RBC proteome by MS in this work, we did not detect actin due to the otherwise mild detergent conditions used to preserve molecular assemblies. Likewise, additional proteins that require strong ionic detergents to dissolve could potentially be missed. We note that membrane protein complexes dynamically exist in multiple forms, as evident from our work in the recently published structures of the band 3 ankyrin 1 complex, Valls et al. 2022, Xia et al. 2022. Another example is the band 4.1 and spectrin complex, which form stable interactions in the absence of actin. The identities of membrane protein complexes depend on purification methods, salt concentration, pH, and so on, and thus the structures of the complexes presented in this work were examined in the context of mild and non-ionic detergents and in the absence of actin. Finally, in defining the RBC interactome, we opted to maximize the quality of the interactions rather than coverage. Therefore, some real interactions will necessarily be absent in the current interactome map. Resource availability.